Welcome to video 5 of IB Biology Topic 3, Genetics. In this video, we are going to be looking at genetic modification and biotechnology. Now, the essential idea behind this unit is that biologists have always been able to manipulate species by trying to increase the possibility of certain traits being passed on. And we've looked at that in artificial selection. Now we're actually looking at manipulating the DNA to pass on specific traits. Okay, so we're looking right down at a DNA level now and a genetic level. In terms of our nature of science behind this area, there is quite a lot of important decisions and ethical decisions that have to be made here. So especially where we're assessing risk in terms of what the implications could be around genetically modified crops and livestock. Okay, so first of all, we're going to just think back to the basics, okay, and that is the concept of this universal code, that all living organisms are made up of the same bases. It is the order of those bases that change genes and therefore change traits. Okay, every codon, each 3 bases code one amino acid, and that amino acid is the same regardless of the species. So the sequence of amino acids in a polypeptide chain remains unchanged depending on the base sequence, depending on the species, but the actual bases involved making the polypeptide chain is the same for all organisms. So it is therefore possible to take genes from one species and insert them into the genome of another, because the actual structure of the DNA is the same. So we call this gene transfer, and it's why our DNA can be called this universal code. We're now going to have a look at the understanding section of 3.5. And in this section, we're going to be looking at 3 different techniques. We've got our gel electrophoresis, and this is where DNA fragments are sorted depending on their length. We've got our PCR, which is where we actually replicate sections of DNA. And we've also got DNA profiling, and that's where we've got our DNA sorted by electrogelphoresis, and we can compare it to other DNA samples. Let's look at those in a little bit more depth. So with our gel electrophoresis, it's all about sorting sections of DNA into size using an electric charge. And you can see in the diagrams the process that occurs. So fragments of DNAs are loaded into wells on a gel, and if you look at the bottom picture, imagine that the wells are running along the top. The gel is then exposed to an electric field, an electric current, with the negative end at the end of the wells and the positive end at the opposite end. So the negative end is the end of the wells and the positive end is the opposite end. As the DNA has a net negative charge, the fragments will then move towards the positive, so opposites attract. Okay, the DNA is pulled towards the positive pole, so the positive end. The shortest fragments will move further and quickest, okay, compared to the longest pieces. That's because they're lighter, they're easier for them to move through the gel, okay. Those of the same length will move at the same speed. The end result is a series of parallel bands of DNA fragments, and all of them at different distances in the gel. So that then gives you a profile that you can compare to other DNA samples. Okay, next up, looking at the polymerase chain reaction, so what we call PCR, and we have looked at this in a previous unit. So this is just a bit of a revision. Okay, so PCR is used to amplify small amounts of DNA. It's used to replicate DNA, and you can see that in the picture. You've got your template, and first cycle, we end up with double the amount. Second cycle, we've ended up with 4 times the amount. These samples can then be used for DNA profiling, recombination, species identification, or any other research. Okay, basically PCR mimics the DNA replication process. The process requires what we call a thermal cycler. It also requires primers, free DNA nucleotides, and DNA polymerase, and we'll look at some of those in a bit more detail soon. Each cycle takes about 5 minutes, and 5 million copies of minute pieces of DNA can be achieved in 5 minutes. So it's a pretty impressive process. Okay, so that was all revision from previously. Now, as we said, PCR occurs in what we call this thermal cycler and involves repeat procedure of 3 steps. So we've spoken about this before. We've got that denaturation, okay, and that's where the DNA gets heated by the thermal cycler, which denatures those hydrogen bonds that hold the strands together. So basically it's unzipping our DNA, exposing those bases. So you can see that in step 1, we've gone from our double helix to it being unzipped. Secondly, we have the process of annealing. So this is after our unzipped DNA has been cooled. DNA primers then attach to the opposite ends of the target genes. Okay, so you can see your primers, you've got your reverse primer and your forward primer moving along. And then in elongation, which is step 3 there, you can see it's called extension on the diagram, but we'll call it elongation. The heat is then increased so that a heat-tolerant DNA polymerase, known as a TUC, replicates the DNA using complementary base pairing. The cycle's repeated over and over, so you get lots of copies. Okay, so we've looked at gel electrophoresis, we've looked at PCR. Now let's just have a brief look at DNA profiling. So we've got our sample of DNA that's been sourced. It could have come from a crime scene, a fossil. Okay, what we now need to do is copy the gene sequences using PCR. So the copied DNA is split into sections using what we call restriction endonuclease, and we'll have a further look at those shortly. Gel electrophoresis is then used to separate out the fragments, and this gives us what we call our DNA profile, which can then be compared to other DNA profiles. Alrighty, so we've looked at those 3 processes, our gel electrophoresis, our PCR, and that idea of genetic profiling. So we will come back to them later in the video. Now that we know that we can identify genes, we now need to look at this whole concept of genetic modification and the manipulation of genes to increase the occurrence of a trait. So if we've identified a gene that, I don't know, the gene that controls red hair, we can take that gene and put it into other genes. So replace the gene so we could increase the number of redheads if we wanted to. Now, before we get to that, we need to understand this whole concept of these restriction enzymes. So we did mention them previously there. Let's have a look at them in a little bit more depth. So restriction enzymes are naturally occurring in bacteria, and they're used in the fight against viruses. Each restriction enzyme has its own deriving from the bacteria from what it was isolated. So it could be a HAP2, a BGL1, it just depends where it's come from. They do not cut at random, but at a recognition site or a recognition sequence. Okay, so it's all around the sequencing of the amino acids. So it's specific sequence of bases on the DNA strand for them, and you'll see that you can get 2 different kinds of cut caused by these restriction enzymes. You can get what we call the sticky ends, so that's at the top there, that's with the piece poking out, compared to the blunt ends, which is completely flat. Okay, so these are blunt ends, whereas this is what we call a sticky end. So when the restriction enzyme cuts the DNA, okay, to break cut the gene out as such, you can have a sticky end or a blunt end. Okay, so looking at genetic modification and gene transfer. The target gene is extracted from the donor in the form of mRNA. Okay, so first of all, we're identifying that required trait. Then we're actually going to extract the target gene, which has been done here in step 3 in the diagram. Reverse transcriptase, okay, and free nucleotides are used to catalyze the complementary DNA strand to form what we call cDNA, so copy DNA. Now, a restriction enzyme, like we just spoke about, is used to cleave, so cleaving means to cut, the cDNA, resulting in this case in some sticky ends. Then what we do, so we've got the piece of DNA, okay, that we want out of the donor gene as such. So from the donor organism, we've got the piece of DNA we want to implant somewhere else. Now what we need is we need a plasmid from a bacterial cell, okay. Now what we do, we cut the plasmid, okay, with the same restriction enzyme because we need to get the same sticky ends and the same complementary base pairs so that we can put the donor gene into the plasmid. So the pieces of the DNA from the two organisms are mixed. Some of the pieces will join the complementary sticky ends of the plasmid. So remember to start off with what we did was we used PCR. We got one gene, we used PCR to replicate. So we've got lots of pieces of the gene that we want. So we're going to put it in with some plasmid. Some of that is going to join into that plasmid, reforming the plasmid's circular shape. Now, DNA's ligase is used to splice the pieces back together. So it seals the new gene into the plasmid, okay, reforming those sugar phosphate bonds. Now, we call this our recombinant plasmid. Okay, so this has been formed and contains that target gene. The recombinant plasmid is then inserted or taken up by the host cell, just like a bacterial cell would be taken up. Host cells then undergo transcription and translation to produce the protein coded for by that inserted gene. So here we've got that in its pictorial form. Okay, so we've got our plasmid, we've got our DNA, and this is humans, we're going to be using the insulin gene. Okay, so we've taken that insulin gene, we've copied it lots of times so that there's more chance of a gene fitting into the plasmid. Okay, so one of the genes that's floating around by the plasmid fits in. Okay, and our DNA ligase has joined that all up. That is then put into what we call our transgenic bacterium, okay, with the plasmid containing that insulin gene, and then they reproduce and reproduce. Alrighty, so we've looked a bit at that genetic modification in the process there, which we will look at further in class. Now we just want to think about this process of cloning. Now, in terms of cloning, we get two genetically identical organisms. Okay, now we can have natural cloning. Okay, that's due to binary fission in our prokaryotes. We've got our plants with buddings and cuttings and sending out runners. Animals in the cases of identical twins. But we can also now, with these advances in technology that we've just looked at, we can also artificially clone. And there's two different ways we can artificially clone: by embryonic division, okay, so forcing twins basically to occur, and also by somatic cell nuclear transfer. So first of all, natural cloning, we're going to look at each of those processes. So we've got binary fission, okay, and we can see that process here where the DNA replicates and then splits into the two daughter cells. So quite a basic process in terms of that compared to what you've looked at. Cuttings and budding, this is where and a second plant grows from the parent plant, genetically identical, so therefore is a clone. We've also got some more pictures of that because you will hopefully be doing a prac where you do some cuttings. Okay, natural cloning, identical twins. So when you get a fertilized gamete and it then splits at that stage, so identical genetics, and the offspring share that placenta. Okay, so now we move on to looking at the artificial clone, that whole concept of forcing embryonic division to occur. Okay, so that's our first type of artificial cloning. And then we've got that whole concept of somatic cell nuclear transfer. So we looked at this in our first unit with the stem cell biology, also known as that therapeutic cloning. Now we will look at these processes further, for example, the somatic cells, looking at how Dolly the sheep was formed, okay, and that's where they took cells from the udder of one ewe and gamete from another ewe and took out the nucleus, and into the nucleus they put the full DNA from the donor ewe, and from that they managed to develop Dolly the sheep. So that is a type of therapeutic cloning. Alrighty, so moving on now to your applications and skills. First of all, the analysis of DNA profiles. Okay, so we've talked about how we set DNA profiles up. We get the piece of DNA, we then go through PCR to replicate it. We then run gel electrophoresis to separate out the DNA, and that then gives us our DNA profile, as we can see on the left. So the DNA profile on the left has come from a crime scene, and what we do is we look along our suspects, and basically we want to see as many matches to the lines on the crime scene as we can to match who our suspect is. And in this case, our suspect would be suspect 2. This can also be used in terms of looking at paternity. Okay, so in this case, we have got the standard, which is the child. Now, what we're looking at is who shares the most genetic information with the child, because you've got to remember the child will have a mixture of the mother's and father's DNA. So what we're doing then is comparing the standard to A, B, C, and D, and we need to work out, hang on, in this case, the percentage of a child is under question. Okay, so we're looking at B, the child, and we need to see whether C or D is its father. So if we're looking at B, okay, each of the child's genes need to match a mother or a father. So the first one matches C. The second one, well, that matches the mother. The third one matches C, and the fourth one matches the mother. So in that case, the father would be C. Got the same thing again here. We've got the child. Second column, we've got the mother to the left, and we've got 3 that could be the father. So if we look down the DNA profile of the child, that one matches the mother, that one matches 1 and 2, that one matches the mother, that one matches number 1, that one matches 1 and 2, that one matches no one, so that could be some kind of mutation. That one matches the mum, that one matches number 1, so I would be saying that number 1 would be the father in that case. Alrighty, so your next application understanding is that gene transfer to bacteria using the plasmids makes use of restriction endonucleocase and DNA ligase. So we've already spoken about how restriction endonucleocase and DNA ligase are used in this process, but let's look at that in a bit more detail. Right, just going back to there. Alrighty, so here we can see our restriction endonucleocase. Okay, working along and reading the base sequence. Okay, now DNA ligase joins the DNA back up. So, excuse me, restriction endonucleocase unzips and DNA ligase reads and rezips. Our next application is the assessment of potential risks and benefits associated with the genetic modification of crops. Okay, so with each of these factors, we've got our risks and our benefits. So first up, if we're thinking genetically modified crops, we've got our environmental factor. So the risks are that non-target organisms could be affected by toxins. So if you are modifying a crop to be resistant to certain types of insects, okay, what you've put in there could then be toxic to other insects as well. The genes could spread, so you could end up with some herbicide resistance. You could get cross-pollination from your GMO crops and other crops, causing such things as herbicide resistance. Then you can't kill volunteer crops that grow in amongst other crops. You could end up with a reduction in biodiversity, because if in your paddock you're only growing cotton, and you've ensured that because the cotton's growing there, you can then spray, say, Roundup, and it kills everything else in the paddock, then you're actually reducing the biodiversity in that area. Benefits of our GMO crops: we actually have to use less insecticide if you have crops that are pest resistant. So you can put a gene in that makes the crop pest resistant, which we will look at shortly. Okay, so the pests then don't affect the crop, which is great. You don't need to spray less chemicals. There's less need for land prep. So if you're going to plant a crop that is not affected by herbicides that would usually kill all crops, what you can do is, as your crop starts growing, you can then go out and spray with herbicides. So what you've done is you can kill any other weeds, your crop keeps growing, and you've got rid of the need to treat the land beforehand. You don't need to plough the land, you don't need to do any other land operations, because you can plant your crop and then you can just spray for volunteer weeds and get rid of them. Genetic modification also allows you to increase shelf life, therefore reducing waste from supermarkets and the like. Okay, so now we'll have a look at through the health aspect and risks involved with GMO crops around health include we're forming these new proteins, so they could cause allergic reactions in people. We don't know the effects because they're new. Potentially the risk of antibiotic resistant genes, which have been used as markers, could then spread into bacteria, and we could end up with issues around the superbugs and things like that we've spoken about before. Those transferred genes could potentially mutate quicker. Again, we don't know. It's uncharted territory, some of this. Benefits for health: there's increased nutritional value in some GMO crops. We can alter them to have more certain vitamins and minerals in higher amounts and things like that. It's possible that we can reduce certain allergens that cause anaphylactic shocks and things like that. And there's potential that we're only just tapping into. So the idea of edible vaccines and things like that, new developments. And then the risks and benefits in terms of the agricultural industry, which is a huge industry in Australia. Okay, so the risks, volunteer crops with herbicide resistance. So if you've got a crop, say you're growing cotton and it's herbicide resistant, okay, which is great because when the cotton's first growing, you can go out, you can spray some Roundup, kills everything else in the paddock, and the cotton keeps growing. Awesome. When you harvest, if you don't manage to harvest all of the crop, or if your harvester ends up depositing some seeds back in the soil, you're then going to get more crops, which we call volunteer. You're going to get these little plantlets growing in the next crop, and you can't kill them, okay, because they're herbicide resistant. So it's a bit of an issue. There is a chance of spreading resistance in insects. So if you have used a specific gene to create some resistance and then insects pick that up, they can then spread that, which could be quite an issue. Farmers can't, when farmers harvest their crops, quite often what they do is they keep a certain amount of their yield and plant it the next year. Okay, so it saves on their costs in terms of sowing their crop every year. That is not allowed with GMO crops. So if you sow GMO crops, you then have to harvest them all and sell them all. And there's specific ways that has to be done, but you cannot keep any to replant. Some of the benefits in terms of agriculture and GMO crops, obviously we can develop crops that are drought resistant, herbicide resistant and disease resistant, all of which increases yield, some of which can reduce the need for dangerous chemicals and pesticides to be sprayed on them. So there are lots of pros and cons to our genetically modified crops, and it's an interesting area to be involved in. Okay, so our next application point is the production of cloned embryos produced by somatic cell and nuclear transfer. Okay, and our example of that is Dolly the sheep, which I spoke about earlier on. Okay, so you take a somatic cell from one plant, you take not plant, we're on animals. You take a somatic cell from one animal, you take a gamete from another animal, same animal, so we're both sheep here. From the gamete you withdraw the nucleus, keep the gamete, you then put the nucleus of the somatic cell into that cell, let it start reproducing, developing, and then you put it into the surrogate. The surrogate you then gives birth to a clone of the parent you where the somatic cell was taken from. A really interesting process. One of your skills is to design an experiment to assess one factor affecting the rooting of stem cuttings. We will get to that in your class time. Okay, now another skill is to be able to analyse data on the risks of monarch butterflies and BT crops. Alrighty, so I did mention that we'd be getting to our BT crops soon. So our BT gene comes from our bacteria there, Bacillus thuringiensis. Okay, there is a gene in that which, once in a crop, means a certain pest, this corn meal, dies. If it eats any part of the plant, it will die if that BT gene is in it. Okay, so what scientists have discovered that if they genetically modify some crops such as corn, cotton with a BT gene, then this bug, or corn borer here that is called, will die if it tries to eat that crop. So BT crops are used all over the world. You'll see there in Australia it's used mainly in cotton. Okay, many countries use it in maize. So maize is sweet corn. All right. So this technology is used all over the world. Now there's a problem, okay, and that is that the larvae of the monarch butterfly also dies if it eats that gene. Okay, so if they feed on the BT crop, then the larvae dies, which means a reduction in the number of monarch butterflies. So basically, by that genetic modification, there is an impact on the biodiversity and the monarch butterfly population. Thank you for watching. There are areas that we will cover in more depth in class, but this is just your introduction.